Hello, everybody. Welcome to the last lecture before your exam. I think Monday is probably going to be a review session. Could be wrong, but it be a review session. But we're going to be going over glycogen metabolism today. There's a lot of slides in this one. I'm going to try and break it down in a way that's not too complicated. A lot of content, but I don't think it's necessary, like I've said for many lectures before. So we will go ahead and get started then. So to start off, we already covered, how this works. I'm not going to go over it again. We already talked about pyruvate kinase, so I'm kind of not. Glycogen. So glycogen is the storage of glucose, basically. Stored in the liver. It's stored in the skeletal muscle a little bit. And what's important to know is that it makes up like 10% of liver, and then 1 or 2% in in the muscle. And the main thing with the liver is that this is exportable glucose. It's not used by the liver, it's exported to other areas. And then with muscle, it's just used within the muscle itself. And then also for the muscle, that those stores are exhausted in a very short time compared to the liver. The whole point of the liver is to store the glucose and then release it to the rest of the body. That is what the liver's job is. So if you see anything to do with releasing of glucose, it's going to have to do with the liver, basically. The glycogen stores look like these little circles, like these like little black dots here. These big ones are not glycogen, but all these little small dots here are just glycogen stores. That you have these mass amounts of glycogen. This is not peptidic glycan. Name for this, I can't remember off the top of my head. Glycogen, I think, is like a polymer starter or primer, if you will, in the middle. So we'll talk about that in a second. So we got glycogen stores in different ways. So you'd think that if you have a lot of glucose in your blood, right, the osmolarity of the blood and of which is kind of messed up, right? Similarly, if you have like a lot of glucose is in your cell, right, it's going to saturate all the water, right, and all the water is going to like surround the glucose and all that jazz. So it's going to be a bit weird. However, the way that we can kind of get around this glucose kind of messing up the osmolarity of your solution is by turning into one massive complex. By turning into one massive complex, we basically take this massive amount of glucose that's all soluble and turn it into this insoluble glycogen molecule that now is no longer really affecting osmotic pressure in any way, right? So basically, it allows the liver and muscles to store glucose without affecting on osmotic pressure. Because remember, osmotic pressure is dependent on the number of solutes dissolved in your solvent, and in this case, glycogen is actually insoluble and actually doesn't dissolve. So basically, taking these 400 millimolar glucose molecules and combining them together to make glycogen is an easy way of making sure your osmotic pressure is not screwed up. Right? These are just the different kinds of solutions. Keep these in mind. You should understand how this works. Hypotonic, meaning that the inside is lower in water than the outside. Hypertonic, meaning the inside is higher in water than the outside. And isotonic, just meaning it's equal. So if we look at glycogen as a structure, we see that the glycogenin dimer serves as a primer. So this G here serves as a primer, right? And then after that, you add these extra secondary primers, and then you basically add these 13 residue glucose branches onto each one of them. So there's 2 branches per chain, right? And that's basically how glycogen is built. So if you look at any of these, you'll see. So you got the primer, and then after the primer, we start looking at actual glucose. So this is the primer, right? If we look at here, this is the first here. There's 2 chains coming off of it, right? If you look at this one here, there's 2 chains coming off of it, right? That basically extends on. So, see here, you got this chain coming off. Basically, it keeps extending out with 2 chains per branch, basically. Or 2 branches per chain, I should say. And there's 2 different types of glycogen bonds, or glycosidic bonds. In these, we got the alpha 1,4 and the alpha 1,6 bonds. The alpha 1,6 bonds are responsible for these called now? Branches, right? And the alpha 1,4 bonds are responsible for the chains. And I think I've talked about this before when I looked at carbohydrates and their structure a little bit. And you should remember reducing and non-reducing ends. The reducing ends have to have an anomeric carbon, so a hemiacetal carbon. There's none of that on these, right? There's no hemiacetal carbon, right? So none of these other ends here are reducing. Plant starch is different. Amylose is not branched. Amylopectin is branched every 24 to 30 residues. So, these are things to keep in mind, but I think there's other things that are important. For glycogen, it's every 8 to 14 residues, so it's a lot more branched in glycogen than it is in amylopectin. Or amylose, because amylose is not branched at all. So now we look at the different, so we've got glycogenesis and glycogenolysis. Now, if you think about this, you try and intuit your way through it, you should be able to tell me that insulin, right? If insulin is for telling you to store glucose, then you can imagine that insulin would lead to the buildup of glycogen. But then glucagon, right, is in the starved state. So you want to produce glucose and send it out to the rest of your body. So you can imagine then glycogenolysis, the breakdown of glucose, glucose, gen, new-ish, and then all the side, like lysis is break, right? So you got new glucose storage breakdown is basically what this means. If you want to think about it. And we've got two different enzymes that are being used. We've got glycogen phosphorylase and debranching enzyme. And debranching enzyme actually has two different functions. It's got a transferase function, if I'm not mistaken, and it's got a glucosidase function. We'll talk about those later. But if you look at this, right, the glycogen has this kind of shape to it, right? It's got these non-reducing ends, and basically what happens is glycogen phosphorylase chops up these glucose pieces, and eventually it reaches a limit, though, where it can't chop it up anymore. Then you get this transferase activity of the debranching enzyme, which takes these three glucoses here and adds them here, and then the glucosidase activity of the debranching enzyme removes this red one here. Then you just have this 1,4 polymer, which you can do this whole thing again here with glycogen phosphorylase with. So, glycogen phosphorylase, how does it work? Well, it works by adding a phosphate. Great. There's really not much else to it. So this basically takes an inorganic phosphate. That's the important thing. Not ATP, inorganic phosphate. This phosphate, basically you can think of it as like it attacks here, this goes back. That how the mechanism works? Probably not, but that's good enough for this class. You don't need to know the ins and outs of how that work. But now we've basically got left with glucose 1-phosphate. Okay. So now with glucose 1-phosphate, that is now going to be isomerized into glucose 6-phosphate with a phosphoglucose isomerase, and then we can go from there. And you can continue doing that over and over again, right, until you get to the point where that enzyme can no longer function. Here is the mechanism if you want to see how this works. I personally don't think this is very important for, the class, but yeah, you can think of it like it's kind of like an E1 reaction, or not E1. E1. Kind of like an SN1 reaction, right? Where you form this kind of carbocation thing, and then you get an attack of the phosphate group. See here, this kind of oxygen here forms a bond here. This ends up leaving, and then this O here comes and attacks there, right? This is when I first saw this, I was like, what is going on? But yeah, so basically right here, right, the limit, right, for glycogen phosphorylase is when it reaches 4 units from a branch. So it'll chop away at this glucose branch until there are 4 things left. So right here, 4 of them. See, there's 4 of these glucose molecules left, and it is no longer chopping away. So once it has gotten to this point, we need to use the debranching enzyme, and specifically the transferase activity, to transfer these 3 yellows over here. And remember, glycogen phosphorylase, whenever it works, it chops up the glycogen and puts a glucose 1-phosphate. The glucose puts a phosphate group, an inorganic phosphate group, on the first carbon of glucose. That can be isomerized into glucose 6-phosphate and go through glycolysis. So this transferase activity, right, puts these yellow glucose molecules on the end here, and then glycogen phosphorylase can actually go back and actually start chopping those up as well. Now we've talked about the transferase activity of the debranching enzyme. Now we have to talk about the glucosidase activity of the debranching enzyme. The glucosidase activity of the debranching enzyme, right, is responsible for cleaving these alpha 1,6 bonds. This bond right here, right? And all there is to that reaction is it's just a hydrolysis reaction. You're using water to cleave that bond, and you're just left with a normal glucose molecule and the rest of your glycogen molecule, right? So normal glucose. So remember, for glycogen phosphorylase, you actually make G1P, but if you look at the debranching enzyme with glucosidase activity, it's actually just a normal glucose. So you actually have to phosphorylate this glucose again to get to G6P and go through glycolysis. So it's a little bit of an inefficiency there, right? But it doesn't really make too much of a difference. But they can totally ask you questions about, oh, you get rid of 4 branches of glycogen. What are your products or something, right? And you'd have to say that there are 4 normal glucose molecules plus whatever else is there. Here we go, phosphoglucomutase, like I talked about, interconverts G1P to G6. All this is what I talked about before. You don't have to know the ins and outs of this, in my opinion, but you just got to know that all the enzyme is doing, right, is that it's basically just adding this phosphate onto the 6th carbon, removing the phosphate from the 1st carbon. And, like I said before, different tissues have different fates of G6P. In muscles, generally what happens is that, it's good that G6P is going to go through it's going to go through glycolysis and citric acid cycle, electron transport chain to make ATP. And we haven't talked about the pentose phosphate pathway, but that is another option that is available. In the liver, though, it just gets dephosphorylated, turned into glucose, and it's exported. Right? So the liver, again, the whole point of the liver is to release glucose into the environment. I hope you're getting this point. I'm trying to hammer this home, right? Releasing glucose into the blood, and the muscles end up using it instead. So hopefully that makes sense. Now, if we look at glycogen, right, we've got the different kinds of methods that this works. So glycogen, right, starting from glycogen, right, we use glycogen phosphorylase to generate a glucose 1-phosphate, right? And the rest of the glycogen kind of just goes over there somewhere. We use a phosphoglucomutase to turn it into glucose 6-phosphate, right? And this can either go to the pentose phosphate pathway to make ribose and ADPH, which we will not talk about right now. Or if it's in the muscle or the brain, right, it gets turned into glycolysis, it goes through glycolysis, turn into pyruvate, and then it turns into lactate, right? Or if it's in the liver, it goes through glucose 6-phosphatase, which is not present in the muscle, and this is an enzyme that's not present in the muscle. And then that turns into glucose and is being used for other tissue. So we have to look at, you'll be wondering, why is it favorable to add just an inorganic phosphate onto a group? I remember, wasn't it unfavorable to add phosphate group onto step 6 of glycolysis with glyceraldehyde 3-phosphate? And you're right, it is actually kind of unfavorable. But the thing is, if you look at the standard conditions, it is unfavorable. However, the ratio of G1P to PI is so heavily favored towards the inorganic phosphate that it actually causes it to be very exergonic. So this Q value is so much less than KEQ that, if you do this delta G here, this is going to be like some value. I don't know. Let's just say that it's +3, right? Like he said. But this is going to be this Q value is going to be so skewed, like 1 to 100, right? And this is, I'm trying to do this math here, but the log of 0.01, right, is -2, and then this is going to be like -10, right? So it's so skewed towards having inorganic phosphate that this overall becomes a -7 kilojoule reaction. But I just made this number up, by the way, this -10, but that's if I plug this 1 over to 100 ratio that they're talking about in there, right? So it's so favorable because of the concentrations of G1P versus the inorganic phosphate. Now we're going to talk about glycogen synthesis, right? So there's a couple steps here to keep in mind for glycogen synthesis, right? We have to activate, so we first have to activate glucose, right? How are you going to make a polymer of glucose if it doesn't want to be turned into a polymer? We first have to activate glucose using UDP glucose, which is just UTP, which is uridine triphosphate, right? Then we have to transfer the glucose from UDP glucose to a growing glycogen chain, and then we have to make these alpha 1,6 branches. So we have to make alpha 1,4 chains and alpha 1,6 branches. So how do we do that? So for to make glycogen, right, we first have to take our glucose 6-phosphate and then we have to isomerize it into G1P, right? That's one of the first steps we do. From G1P, right, we then take our UTP and we attack it with our G1P. And you're probably thinking, well, why is that favorable? Well, we'll talk about that in a second. It's actually listed over here if you look at this, but we'll talk about it in a second, right? So after we form that, right, this PPI is released, right? And now we're left with this molecule, this UDP glucose molecule. And the reason this reaction is favorable, actually, so if we look at this, actually, the UDP glucose pyrophosphorylase, this kind of addition of UDP onto UDP glucose is actually not, there's actually no real delta G difference there because we're going from like a phosphodiester bond into another phosphodiester bond, right? It doesn't matter. It's like if you look at this is the same thing as pretty much this. It's a little bit different, but it's pretty much the same. So the delta G is pretty much 0 there. However, what makes this reaction favorable is the fact that it's coupled to the hydrolysis of pyrophosphate. And the hydrolysis of pyrophosphate has a delta G of negative 19 kilojoules. It is very stable to hydrolyze this pyrophosphate. So that is why that reaction is actually very favorable, right? It's because it's coupled, and because this pyrophosphate hydrolyzation is actually what makes it favorable. So the question is, where does this glucose 1-phosphate come from? And just like I said before, probably from an isomerization reaction of G6P, right? And then what other modifications do you know they're used to activate metabolism? Yeah, I mean, phosphorylations, right, phosphorylation is like one of the big ones. You can also do things like carboxylate things. Like you can remember, if you remember how it worked for pep carboxykinase, right? We turned it into oxaloacetate and then used that carbon dioxide molecule to kind of turn it into a good nucleophile to grab a phosphate. So there's many different ways you can activate things and turn them into reactive compounds. So now we have this UDP glucose, right? So now we have to figure out how do we what do we use to attack it? So if we look at, if they mention the enzyme here, but it doesn't look like it. It does talk about pyrophosphatase, but yeah, if we just look here, the way that we basically make this a molecule here is I just want to double check here for, yeah, I thought they did mention this up here. Did they not mention it? Okay, so oh yeah, here we go. Yeah, they did. Okay, yeah, for some reason I forgot about this. Yeah, so the enzyme that does this whole process of adding UDP to glucose, right, is UDP glucose phosphorylase, right? And then pyrophosphatase is also coupled with this reaction here to make it negative delta G, right? So this is, for glycogen synthesis, this is one of the enzymes and this is the other enzyme, but this is the main enzyme you should keep in mind, pathway. If we move down here, right, now we're on to the next enzyme, and what is now using glycogen synthase. So glycogen synthase is a little bit different from UDP glucose pyrophosphorylase, right? In the sense that now that we have our activated intermediate, we have to use it to make glycogen. And the way that is done, right, is that we basically take this UDP glucose, right? It's in an activated form now, right? And basically what's going to happen is this O is going to basically attack here. And this is going to, these electrons are going to come back to this O, right? And you basically release UDP as a byproduct. Now we form this alpha 1,4 linkage, right, between glycogen and this incoming And here we've got the branching enzyme, right? So once you've made these chains using the glycogen synthase, right? You got to use the branching enzyme. We had the debranching enzyme. Now we have the branching enzyme, right? The glycogen branching enzyme, right, is going to be used to take these chains that you've added, right? So you using glycogen synthase, you make this really long chain, right? And then using the the branching enzyme, you basically take one of these bonds and the chain with it, and you basically attach it on top of this glucose. And I don't think you need to know the exact mechanism for how this works, right? It's just important to know how, like, that this is how, like, this is the enzyme that actually does that branch formation, right? It takes these alpha 1,4 chains, turns it into alpha 1,6 branches, and then from there you can keep doing this process over and over again with glycogen synthase and glycogen branching enzymes. So the way that this all starts has to do with glycogenin, which is a glycosyltransferase, and it's basically an autocatalyzing enzyme. Um, it basically just makes its own. It makes its own glycogen, which is how you actually start making glycogen, glycogen enzyme, like glycogen storages, right? So the enzyme basically acts as its own substrate. So basically, the enzyme has a catalytic domain, right, and then it's itself kind of fits into its own catalytic domain, and then it basically gets turned into a glycogen primer. So here is the example of that. So if we look at this, right, you've got glycogenin, right? And it's basically an autocatalytic formation here. So you form a glycosidic bond between glucose and the tyrosine of glycogenin. So the tyrosine acts as a nucleophile, right? So this glycogenin, right, is attached to a tyrosine, and this tyrosine forms a bond with this glucose molecule and releases UDP in the process, right? So this is a glucosyltransferase activity, right? And then you basically just continue doing this process over and over again, where you keep adding UDP glucoses until you just get continuous chain extension, right? So this second reaction, right, the addition of like 7 more glucose molecules is done by glycogen synthase. But this first reaction, right, has to be done by glucosyltransferase activity, right, from glycogenin. So the glycogenin's glucosyltransferase activity is what primes the glycogenin by adding the first glucose. But then everything after that, so everything from here downwards, right, the is using a glycogen synthase, right? And we talked about this already. I'm not going to go over this again, but if you want to take a look at it, go ahead. So now we have to talk about regulation, right? So we talked about glycogen synthase, which stores glucose, and glycogen phosphorylase, which releases glucose. These are the main enzymes that are regulated in this process. The other enzymes are probably also regulated a little bit, but for the sake of this class, these are the two enzymes that you can think about as being regulated in this process. So they're both regulated allosterically and covalently, right? And then there's covalent modifications under hormonal control. So you should immediately be thinking about insulin and glucagon, phosphorylation, dephosphorylation, right? And then there's different isozymes of the phosphorylase in muscle and liver, but don't really care too much about that. So let's think about this, right? So even if I don't look at this, right, you should be able to figure this out on your own. So glucagon is a low glucose, right, which leads to glucose release, right? And which enzymes promote glucose release, and which ones promote glucose storage. So let's think about this, right? So glycogen phosphorylase, right, promotes glucose release, and glycogen synthase promotes glucose storage. But glucagon, we know, if you remember from our pathways, glucagon causes phosphorylation, right, through protein kinase A, right? So if glucagon causes phosphorylation, right, then we should expect that phosphorylation activates glycogen phosphorylase and turns off glycogen synthase, right? So glucagon activates phosphorylase in the liver, right? Epinephrine, a similar story, right? Fight or flight, right? Leads to glucose release. We would expect it to, similar to glucagon, lead to an activation of phosphorylase, which is a phosphorylation of this enzyme, right? Insulin, on the other hand, right, is usually leads to dephosphorylation, right? So insulin leads to dephosphorylation. If you think about glycogen phosphorylase, if it's active in its phosphorylated state, it must be inactive in its dephosphorylated state, right? For glycogen synthase, right, it must be active in its dephosphorylated state and inactive in its phosphorylated state. Hopefully you can see how I'm doing this, right? I'm basically saying, okay, insulin leads to glucose storage. Which enzyme leads to glucose storage? Glycogen synthase leads to glucose storage. Insulin causes dephosphorylation. Therefore, glycogen synthase must be active in its dephosphorylated state, right? You can kind of see how I'm like using my prior knowledge to kind of infirm or like kind of work my way through it, even if I didn't memorize the exact phosphorylation, dephosphorylation states of all these enzymes. You shouldn't have to. You should be able to use that general rule of thumb of glucagon equals phosphorylate and insulin equals dephosphorylate to figure out what state enzymes are active or inactive in. Right? So so this is a very intimidating slide. I wouldn't worry about this. Basically, what this is trying to say is that glucagon, right, leads to phosphorylation through protein kinase A, and it's a very exponential pathway, right? So glucagon leads to cyclic AMP, right? Adenylate cyclase, and that leads to a bunch of pKa activation. And that pKa activation is basically what the pKa activation, right, is essentially what the PK activation is basically how we get this phosphorylation cascade for glucagon, and it's also why we assign glucagon equals phosphorylate, basically. So yeah, an enzyme cascade is basically just this idea that one thing leads to another, which leads to 20 other things, which leads to 20 other things. It becomes exponential, right? Signal cascades, right? Hopefully you remember that from Bio 220, right? But yeah, and if you look at here, right? Surprise, surprise, we just talked about this, right? When it's in the dephosphorylated state, phosphorylase is inactive. When it's in the phosphorylated state, it's active, right? That makes sense based off of what glucagon and insulin should be doing, right? I would encourage you to think about these two in your head, right? How do they work? And to try and see if you can go through my line of reasoning there and see if you can get that right. So if we look here, yeah, so basically, right, what is going on here, right, is that if we look at the liver versus the muscle in terms of what is happening, right, when the liver receives signals from glucagon and epinephrine, right, it basically upregulates glycogenolysis and gluconeogenesis. So pyruvate and glycogen get turned into glucose 6-phosphate and it's released into the blood, right? When epinephrine hits the muscle tissue, though, right, we get this different thing where instead of promoting gluconeogenesis, right, because you don't do gluconeogenesis in the muscle because they don't have a lot of those enzymes, they don't have glucose 6-phosphatase, right? Instead, we would probably inhibit it actually, and we would actually promote glycolysis and glycogenolysis, right? So these the glycogen would be used to break down into glucose 6-phosphate and then turn into pyruvate, which is very different from the liver because the liver turns the pyruvate back into glucose 6-phosphate to send it up to the rest of the blood or the rest of the cells, right? So this arrow is actually flipped. These two, right? This other thing I want to kind of check is glucagon acting on muscle tissue, and it does. See, for some reason it doesn't show that, but glucagon does actually act on muscle tissue from what I understand. So you should also keep that in mind. So, lo and behold, we've actually already talked about this. Do you have to know this exact mechanism for this? No, probably not. Is it helpful to know? Sure, but I wouldn't say it's the most important thing to memorize. We already talked about this though. So phosphoprotein phosphatase, right, removes phosphate groups. What would trigger this to work? Insulin. Insulin triggers dephosphorylation, right? And lo and behold, PP1 is activated by insulin, right? And the opposite, though, is the kinase. The kinase would be stimulated by glucagon and epinephrine, right? So you could see how I'm able to predict these before it even shows up in these like slides, right? So you look at this, right? Glucagon, right, would trigger a phosphorylation, which is a kinase, which would turn it into the active form because it's a phosphorylase, right? The phosphorylase is used to release glucose, right? And glucagon's job is to release glucose, right? The other thing that turns this on is high levels of AMP and calcium. A calcium, right, is calcium's a bit of a weird one. You have to kind of memorize it, but if you remember from BIOC 20, calcium release means muscle contraction, right? And so you can think of it like that. If there's a lot of calcium in your cell, it means that your muscles are contracting a lot, so you're going to want a lot of glucose release, right? And then AMP is just AMP, right? If you have high AMP, it means you have lower ATP, right? If you have lower ATP, it means you need more energy, and the way you do that is by using glycogen phosphorylase, right, to break down glycogen, right? So it makes sense, right, that in the phosphorylated form, it would be the active form, right? And in the dephosphorylated form, right, it would be the inactive form, right? And that's what insulin does with protein phosphatase 1. Insulin turns off the glycogen phosphorylase. Right? And if we go down here, my throat is killing me, man. If we go down here, right, we also see that liver glycogen phosphorylase is a glucose sensor, right? And glucose actually deactivates the phosphorylase in the liver, which makes sense, right? If you have a lot of glucose, you don't want to keep breaking down glucose, right? So there actually are two ways to kind of stop this enzyme from working. You could dephosphorylate it, or you can bind glucose to it, right? So actually what happens is that the binding of glucose actually makes the enzyme more susceptible to dephosphorylation by insulin, because otherwise these groups actually stay hidden within the enzyme itself, right? So you can think of it like when glycogen phosphorylase is active, right, in its phosphorylated state, right, it's going to keep breaking down glycogen, right, and releasing glucose, right? Eventually, right, that glucose will feed back onto glycogen phosphorylase, right, and fill these two sites here. And these two sites being filled by glucose leads to a conformational change where these phosphates are now exposed so that protein phosphatase, right, that is activated by insulin, right, can dephosphorylate these groups to remove the phosphates and put it in its inactive form, right? So basically we're saying it's a glucose sensor because it's able to bind glucose in these two slots, right? That makes sense? It's being able to bind glucose is why it acts as a glucose sensor. And, we talked about glycogen phosphorylase right here. Let's talk about glycogen synthase as well. But this is really just the same deal, right? There's really not, calmodulin is a bit of a weird one. So you guys haven't learned about calmodulin, but what you should know is that calmodulin is actually calcium regulated, right? So it's actually, just think of it like a calcium dependent kinase. So when calcium binds to it, right, it actually leads to activation of that uh, activation of that kinase, basically, how you should think of it. So, right, if we look at glycogen synthase, right, we can stimulate protein phosphatase, right, to induce activity of glycogen synthase, right, because insulin promotes activity of glycogen synthase, right, and insulin promotes phosphatase activity, right, and since we're trying to promote the storage of glycogen, right, that means that glycogen synthase, which promotes storage of glycogen, must be active in its dephosphorylated state, right? So, if you think about this, right, epinephrine and glucagon, right, these all lead to phosphorylation, right? They also suggest breakdown of glucose, but that's not what glycogen synthase does. Glycogen synthase promotes buildup of glycogen, right? But since these are promoting the opposite, right, that means that glucagon, which leads to phosphorylation, right, must inactivate glycogen synthase. So phosphorylation leads to inactivation of glycogen synthase, which is the opposite of glycogen phosphorylase, right? Glycogen phosphorylase, you phosphorylate glycogen phosphorylase, it actually leads to activation of it, right? You have the R state and the T state, right? So dephosphorylation leads to the R state of this enzyme, and phosphorylation leads to the T state, right? So, with that in mind, right, we can say that all of these kinases, right, lead to the inactivation of glycogen synthase, right? And using insulin, dephosphorylation, right, using insulin leads to activation of glycogen synthase, right? And you can see here, right, how these two things kind of are reciprocal inhibitors in a way, or reciprocal modulators, right? They don't have the same effect. They have the same phosphorylation or dephosphorylation effect on both enzymes, but what that phosphorylation does to the specific enzyme actually changes. So if we look at the glycogen phosphorylase and the glycogen synthase in the presence of insulin, let's actually look at um, let's look at phosphorylase in both scenarios, right? So notice how under insulin conditions, right, this leads to dephosphorylation, which leads to the inactive form of the phosphorylase. For glucagon, right, it phosphorylates the phosphorylase, right, which leads to the active form, right? Something to keep in mind, right? So insulin, right, usually leads to dephosphorylation, and insulin promotes storage. So it would, since it leads to dephosphorylation, right? Dephosphorylation of glycogen phosphorylase leads to the inactive form, and glucagon, which promotes glucose release, right, would activate glycogen phosphorylase, and glucagon causes phosphorylation. So the phosphorylated form of this is the active form. You can do the same thing with glycogen synthase, right? Since glycogen synthase promotes buildup and storage, right, insulin would activate it, so dephosphorylation will activate it. And glucagon, which promotes glucose breakdown and glycogen breakdown, would lead to phosphorylation, which leads to the inactive form, right? So here's the whole thing, right? You've got this whole pathway again, right? I'm not going to really go in depth on this too much because we just talked about it, right? But you should be able to go through and see how all this works, right? So insulin, right? Glycogen synthesis is required, which leads to the activation of this phosphatase, right? And this leads to the inactivation of glycogen phosphorylase because we don't want to break down glucose, we want to build it up. But then it activates glycogen synthase, right? And so glycogen synthase, right, is activated in its dephosphorylated state, right? And so you get this activation that leads to the buildup of glucose and glycogen, right? And this is the idea of reciprocal regulation, like I said, right? Phosphorylase and synthase, they're affected by both glucagon and insulin, but the effects that these two things have on them is inverse, right? So I would basically go through these key points here and trying to understand how this works. But basically, right, for glycogen synthesis, right, you need glycogen synthase, you need that UDP glucose phosphorylase, right, that enzyme, right, UDP glucose pyrophosphorylase, if I'm not mistaken, right. You need that to activate the glucose. You need pyrophosphatase to lead to hydrolyze that pyrophosphate to make this a favorable reaction, right? You need glycogen synthase to actually add the glycogen, the UDP glucose to the growing glycogen chain. You need the branching enzyme, right, to kind of form those alpha 1,6 branches after the 1,4 alpha 1,4 branch alpha 1,4 chains have been formed, right? And then you need to be able to differentiate, the difference between the liver and the muscle, right? We already talked about up in the top, right? How does that work, right? In the sense that glycogen metabolism in the liver, leads to the release of glycogen into the blood, while in the muscle it leads to more leads to just glucose being put into itself, so it can perform more cellular respiration, right? Um, but I should specify that when we talk about like glycogen phosphorylase, specifically it's a liver, right, that is able to sense this glucose. By the way, I should probably specify that, right? So that's kind of the main thing, right? That's different between the liver and the muscle in terms of glycogen phosphorylase, right? In the liver, it's sensitive to glucose like this, while in the muscle, it's not really sensitive to glucose in this sense. And then yeah, we talked about this, right? Glucagon, epinephrine, insulin. I've tried to hammer this point home with how these work. Hopefully, it makes sense because it's very very important to understand how glucagon and insulin work. Glucagon phosphorylation, insulin dephosphorylation, epinephrine also phosphorylation, right? So make sure you try and keep this in mind, right? This whole liver glycogen phosphorylase, the glucose sensor, is important. Um, the fact that this takes part in the liver and not really in the muscle is kind of what separates it from the muscle cell, which is which is important. Similar to how like glucose 6 phosphatase is not present in muscles, but it is in liver, right? So the liver has some slight differences, right, that they could test you on. So just keep those differences in mind and start to study them thoroughly and understand how they work. But yeah, other than that, I hope this video was helpful. It's a little bit fast. I know there's a lot of content to cover here, but I want to try and make sure I'm going through it relatively quickly and trying to get these videos out to you guys as fast as I can. Though I know that I can only do this on the weekends, unfortunately. So yeah, hopefully it was helpful though. Thank you for watching and happy studying. Hope to see you at workshop on Monday.